Hi, my name is Dr. Jamie Caroon. I'm the medical director at the Center for Medical Cannabis Education in San Diego, California. In this video, we are going to look at the first study to show that FDA-approved doses of CBD can lead to elevations in liver enzymes in healthy adults.
This study is entitled Cannabidiol and Abnormal Liver Chemistries in Healthy Adults, Results of a Phase 1 Clinical Trial. It was published in October of 2020 in the journal Clinical Pharmacology and Therapeutics. This study was a Phase 1 open-label drug-drug interaction trial. The study population was comprised of 16 healthy adults, and this is important because these individuals did not have epilepsy, which is the medical condition that the intervention here, Epidiolex, was approved by the FDA in the US to treat. The intervention was 1,500 milligrams per day of CBD in the form of Epidiolex, administered orally in 2 divided doses over approximately 3.5 weeks. There was no comparison drug per se, but caffeine was used as a probe for cytochrome P450 1A2 activity because caffeine is a substrate for that enzyme. Outcomes included liver enzymes, including ALT and others, PT-INR, which is a measure of blood clotting, the association between cytochrome P450 2C19 metabolizer status and ALT levels. CYP2C19 is the major enzyme involved in phase 1 metabolism of CBD. And finally, the association between plasma CBD levels and ALT levels. By way of background, Epidiolex is an FDA-approved drug for 2 rare forms of intractable epilepsy, namely Lennox-Gastaut syndrome and Dravet syndrome. It was approved in June of 2018. It was granted orphan drug status, which means that the drug approval process was expedited, and it was largely approved based on the results of 3 randomized controlled trials, which we will look at in a moment. This drug was initially scheduled by the DEA as a Schedule 5 controlled substance, but in April of 2020, the drug was descheduled. It was developed by Greenwich Biosciences here in the US and GW Pharma in the UK, same company. Greenwich Biosciences is the US subsidiary. And the ingredients are 100 milligrams of CBD extracted from cannabis and highly purified per ml. Also included is dehydrated alcohol, sesame seed oil, strawberry flavor, and sucralose. Here are the 3 trials I mentioned. In these 3 trials, there was a clear dose-dependent relationship between CBD and elevations in liver enzymes, particularly transaminases. And we're going to take a quick look at these 3 studies. This study was published in the New England Journal of Medicine in May of 2017. It was a randomized, double-blind, placebo-controlled trial. It was a phase 3 clinical trial. The population was comprised of 120 participants, all with treatment-resistant Dravet syndrome. Ages ranged from 2 to 18 years of age. It was implemented at 23 different clinical sites around the world, mostly in the United States and Europe. In the intervention group, there were 61 participants. They received 20 milligrams per kilogram of cannabidiol or Epidiolex orally per day for 14 weeks. The 59 participants in the comparison group received a matched placebo. Primary outcome was change in convulsive seizure frequency, and adverse events were also looked at in depth. There was a positive result in terms of the reduction in convulsive seizures, but I'm going to be focusing in this presentation on the adverse events and more specifically on the biomarkers of liver function. Both mild to moderate and serious adverse events were statistically significantly greater in the group receiving Epidiolex, and they were experienced by the vast majority of participants. According to the authors, increases in ALT and AST greater than 3 times the upper limit were observed in 12 out of the 61 participants receiving Epidiolex. That's about 20%. All of these participants were taking valproate concurrently. Valproate is a common anti-epileptic drug. On the right, you can see the number of concurrent anti-epileptic drugs per participant in the Epidiolex group was 3, with a standard deviation of 1. And below that, you can see that the other anti-epileptic drugs are listed. 66% of the participants in the Epidiolex group were taking clobazam, and 61 were taking valproate. The next study was published in The Lancet in March of 2018. It was also a phase 3 randomized, double-blind, placebo-controlled trial. The population was comprised of 171 individuals with treatment-resistant Lennox-Gastaut syndrome. The age of these participants ranged from 2 to 55 years of age. It was implemented at 24 clinical sites internationally. There were 86 participants in the Epidiolex group. They also received 20 milligrams per kilogram of Epidiolex orally per day for 14 weeks, and the 85 participants in the comparison group received a matched placebo. The primary outcome in this trial was change in monthly drop seizures, as well as adverse events. Again, seizures were reduced in the Epidiolex group, and both mild to moderate and severe adverse events were statistically significantly greater in that group. The vast majority of participants also in this study reported adverse events. According to the authors, increases in ALT and AST greater than 3 times the upper limit of normal were observed in 20 participants out of the 86 in the Epidiolex group. That's 23%. 16 of those 20 were concurrently taking valproate. All elevations were reversible after adjusting the dose or discontinuing Epidiolex and or one of the other anticonvulsant drugs. On the right, you can see that the median number of concurrent anti-epileptic drugs per participant was 3 in the Epidiolex group, with a range of 1 to 5. So already we are seeing an association between transaminase elevations and use of anti-epileptic drugs, particularly valproate. This is the third study. It was published in the New England Journal of Medicine in May of 2018. It was also a phase 3 randomized, double-blind, placebo-controlled trial. The study population was comprised of 225 individuals with treatment-resistant Lennox-Gastaut syndrome, age range 2 to 55 years. It was implemented at 30 clinical sites, mostly in Europe and the United States. 149 individuals were in the intervention group. There was a low-dose group and a high-dose group. 73 individuals took 10 milligrams per kilogram of Epidiolex daily for 14 weeks, and 76 individuals took 20 milligrams per kilogram. 76 individuals in the comparison group took a matched placebo. The primary outcome was change in monthly drop seizures, again, and adverse events. Both mild to moderate and severe adverse events were statistically significantly greater in the CBD group and significantly higher in the 20 milligram per kilogram group as compared to the 10 milligram per kilogram group. According to the authors, increases in ALT and AST greater than 3 times the upper limit of normal were observed in 14 out of the 149 participants receiving Epidiolex. That's approximately 8%. And most of them were in the 20 milligram per kilogram group. That's approximately 9%. 11 were taking valproate concurrently. On the right, you can see that the median number of concurrent anti-epileptic drugs per patient was 3 in the 10 milligram group with a range of 1 to 5, and 3 in the 20 milligram Epidiolex group with a range of 0 to 5. Some conclusions can be drawn just from a brief review of these studies. Elevations in liver enzymes, particularly transaminases, seem to be dose-dependent and seem to be related to concurrent administration of other anti-epileptic drugs, and perhaps pre-existing liver conditions which may also be associated with concurrent administration of other anti-epileptic drugs. Moving on, here is the Epidiolex drug monograph. It warns of elevations of transaminases and says that the risk may be greater at higher dosages of CBD, and among those taking CBD and valproate. I assume these warnings came directly from the studies that I just reviewed. And here at the top, the monograph says to obtain serum transaminases and total bilirubin levels in all patients prior to starting treatment. It also offers recommendations for dosage. Here is the maximum recommended maintenance dose of Epidiolex, 20 milligrams per kilogram per day. If we apply the maximum recommended maintenance dose of Epidiolex to the average female in the US, who weighs 170.5 pounds according to the CDC, the daily dose of CBD is 1,550 milligrams per day, which is basically the dose used in this study that we are about to dive into. Now, the FDA is concerned about all of the non-FDA-approved CBD products available at health food stores and grocery stores and gas stations. These products are generally full-spectrum hemp-derived CBD products, and they are widely available. They're also being used by individuals to treat a broad range of symptoms and medical conditions, generally without a consult with a healthcare professional, and without obtaining serum transaminase levels and total bilirubin levels prior to starting. Thankfully, consumers are using markedly lower doses of CBD in these products than those being used in the aforementioned clinical trials, but the FDA is justifiably concerned. The investigators of this study state that these observations suggest that people without baseline liver conditions, not receiving valproate, and taking lower doses of CBD should be at much lower risk of hepatotoxicity than patients with Dravet syndrome and Lennox-Gastaut syndrome. So this tells us something about the investigators' expectations. Okay, now we're going to take a deep dive into this study. In contrast to the previous studies that I briefly reviewed, this study was conducted in healthy subjects who were not on any medications during the treatment period and did not have epilepsy of any form. So this is a very different sample of individuals. The study population was mostly comprised of females in their mid to late 20s, roughly 2/3 were female, and over 80% were Caucasian. Here is a basic overview of the study. The trial was conducted in the Netherlands between April and July of 2019. 16 participants were enrolled on day -1. On day 1, participants took a single dose of 200 milligrams of caffeine. The first dose of CBD was taken on day 3. Participants were instructed to escalate their dose at home until they reached the maintenance dose of 750 milligrams per day on day 12. Blood was collected at ambulatory visits on days 12, 18, and 23, and on a residential visit on day 27, and on follow-up. On day 26, participants took another single 200 milligram dose of caffeine, but this time with their morning dose of CBD. That way, the investigators could determine the impact of CBD on the metabolism of caffeine by CYP1A2. Apparently, those pharmacokinetic results are being published in a future publication. And, according to the Epidiolex drug monograph, substrates of CYP1A2 may also require dose adjustment. Participants were discharged on day 28. Follow-up visits occurred 14 and 16 days after the last dose of Epidiolex. With regard to the results, 14 out of the 16, or 88%, experienced adverse events. The most common were gastrointestinal in nature, and most were first experienced by the participants during the CBD titration phase when they were at home, increasing their dose up to the 1,500 milligram dose. Adverse events were generally mild or moderate in terms of their severity, and 7 of the 16, or 44%, experienced serum ALT elevations greater than the upper limit of normal. 5, or 31%, experienced serum ALT elevations greater than 3 times the upper limit of normal, and were withdrawn from the trial prior to completion. This is Table 2 from the study. It shows the number and percentage in parentheses of participants whose peak ALT was greater than the upper limit of normal, using both the limits set forth by the investigators and consensus limits, which are considerably lower. As you move from left to right, each column represents the same information, but with an increasing multiplier times the upper limit of normal. 3 times the upper limit, 5 times the upper limit, etc. And so here, 7 out of the 16, or 44%, had ALT levels greater than the upper limit of normal based on the stated upper limit of normal, which was 68. If more conservative consensus estimates are used, 11 of the 16, or 69%, experienced ALT elevations greater than the upper limit of normal. And importantly, 5 participants out of the 16 met the international criteria for defining drug-induced liver injury by experiencing ALT elevations greater than 5 times the upper limit of normal. No association was seen between serum ALT elevations, sex, BMI, or genotype. With regards to activity of CYP2C19, metabolizer status was determined by genotype. Of the 7 participants who had ALT elevations greater than the upper limit of normal using the stated upper limit, 2 were considered intermediate metabolizers, 4 were considered extensive metabolizers, and 1 was considered an ultra-rapid metabolizer. Ultra-rapid metabolizers typically have normal or increased enzyme activity, while extensive metabolizers typically have normal enzyme activity, and intermediate metabolizers have intermediate enzyme activity. So you might expect rapid metabolism of CBD to be associated with lower peak levels of ALT, and that seems to be the case for ultra-rapid metabolizers, but not for extensive or intermediate metabolizers. And even here, there's a poor metabolizer that has a peak ALT concentration below the line of 1 times the upper normal limit. So CYP2C19 genotype does not appear to be associated with peak ALT levels. This is Figure 5 from the study. Mean plasma CBD concentration is on the vertical axis, and peak ALT concentration is on the horizontal axis as a multiple of the upper limit of normal. So this is 0.1 times the upper limit of normal. This is 1 time. This is 10 times the upper limit of normal. The red dots are participants in this trial, and the black dots are participants in another published trial in healthy adults that received comparable doses of Epidiolex for greater than 3 weeks. Now you might expect a linear relationship between mean plasma CBD levels and peak ALT levels, but that's not the case. The authors report that there was no correlation. Here you can see that one individual with a mean plasma CBD level of around 100 nanograms per milliliter has maybe 0.2 or 0.3 times the upper limit of normal with regard to their ALT level. Yet these individuals who have roughly the same plasma CBD concentration have ALT levels perhaps 8 or 9 times the upper limit of normal. So this represents a tremendous amount of pharmacokinetic variability, which is typical of pharmacokinetic studies, which look at these highly polymorphic cytochrome P450 enzymes. The authors state that when analyzing data from the current trial and a published trial investigating CBD withdrawal, there was no correlation between systemic exposure to CBD and peak ALT levels. In conclusion, this is the first study, according to the authors, to find that FDA-approved dosages of CBD can lead to elevations in liver enzymes in healthy adults. 7 or 44% of participants experienced ALT values greater than the upper limit of normal using the stated criteria. The number and percentage are higher using consensus upper limits. 5 or 31% experienced ALT values greater than 5 times the upper limit of normal, and therefore met the international criteria for drug-induced liver injury. The number and percentage is higher using the consensus estimates. The mechanism underlying the elevations has not been identified, but it seems clear now that it's not due to concurrent medications. The 3 studies that we looked at earlier in non-healthy adults all showed an association between dose and levels of elevated enzymes, as well as anti-epileptic drugs and levels of enzymes. The mechanism underlying the elevations has not been identified, but it does not appear to be due to concomitant use of other anti-epileptic drugs, since this population wasn't taking any. Elevations were reversible, fortunately, and the authors report that there was no correlation between peak ALT elevations and CYP2C19 metabolizer status, nor peak elevations and mean plasma CBD levels. In closing, I should point out that the study was funded by GW Pharmaceuticals. 2 of the 4 authors were professors from the School of Pharmacy and Institute of Drug Safety Science at UNC, University of North Carolina. They also happen to be paid consultants for GW. The other 2 authors are employees of GW, including the chief medical officer.
With that, I'd like to thank you for your time and your attention. If you have any questions or feedback, please feel free to reach out to me at the email address below.